Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 185 for the podcast. Today, I'm joined by Professor Matthew Wood, a leading figure in neuroscience and RNA-based therapeutics. He is Professor of Neuroscience at the University of Oxford, Deputy Head of the Medical Sciences Division, and Director of both the MD UK Oxford Neuromuscular Centre and the Oxford-Harrington Rare Disease Centre, a groundbreaking partnership between the University of Oxford and Harrington Discovery Institute that's dedicated to accelerating therapies for rare genetic diseases affecting millions worldwide. In today's episode, we discuss his vision for making antisense oligonucleotides, or ASOs, and gene editing more modular, more scalable, and faster by collaborating with regulators, scientists, and patient groups to bring hope to those with rare neuromuscular and genetic conditions. With Rare Disease Day coming up just next week, I hope you enjoy the insights that Professor Wood shares on the future of the fight against rare disease. Matthew, welcome to Beyond Biotech.
Thank you very much, Dylan. It's great to be with you.
Matthew, can you maybe start by just sharing your journey into neuroscience, into rare disease research? How did you get to be exactly where you are?
Yeah, a pleasure. So my background is I'm an MD, PhD in the kind of US parlance. I started off in medicine, studying medicine. I would say I've always been interested in the nervous system and the brain from my earliest days studying medical science. But it kind of dawned on me that a lot of the problems in neurology and neuroscience in particular were just intractable. They were untreatable. And this was in the late 1980s, early 90s. And that clearly, if one wanted to have an impact on that, one needed to get into fundamental research and to think about how one could develop new therapies. And so I ended up moving, because I trained in Cape Town in South Africa in medicine. I moved to Oxford to do a PhD. And that was really the start of my research career. And at the time, in the 1990s, my PhD happened to be working on gene therapy for brain disorders and the early days of gene therapy vectors. And so that's how I got hooked in. And I still believe that there really is a great need for sort of research and R&D. Although there's been huge progress, there's a need to bring bright people in to really drive all the future innovation in medical science, particularly for neurological problems.
Was there something that actually drew you to neurology, though? Is there some feature, somebody you knew, something you read?
Yeah, there was actually. Well, there were a couple of things. The complexity of the brain. I mean, I was interested in complicated things, so I think it was natural, the nervous system and the complexity of the brain. But I had a mentor early in my medical training who was a neurosurgeon, and I recall spending nights just in the surgery theater, watching neurosurgical operations when I was kind of 19 years old. And that was kind of really interesting. He went on from Cape Town at the time to become a professor at UCLA in California, and we kept in touch. And so that was a really first-hand inspiration of how a really skilled neurosurgeon could save people's lives and could repair, neurological problems through surgery. So that was really the initial inspiration. Yeah.
And now you're professor of neuroscience at Oxford, deputy head of the Medical Sciences Division, and you're working in all sorts of things, RNA-based medicines, rare disease. I mean, your day job, your calendar must be book solid.
Yeah, no, it's busy. But I'm trying to cut down and focus. Yes, I think being in Oxford for my PhD led me into the area of muscular dystrophy. At the time, which is a rare muscle and neuromuscular disease. At the time in the 1990s, it was a time when the gene that causes muscular dystrophy had been identified, one of the earliest genes through all the old methods that we used in the 1980s to identify genes. And it became feasible in the 1990s to think about how you might treat this genetic rare disease through either gene therapy, and we could talk about that. There are gene therapies approved now for muscular dystrophy almost 30 years later. But at the time, the first antisense oligonucleotide experiments and potential therapies were tested out in Japan and in the UK and in the US, and that was about 1995. So that really was a part of the motivation for the rare disease work, but it was very, very focused on muscular dystrophy. I have a family member with muscular dystrophy, not a close member, a cousin. So there was some really close family interest as well. But originally it came from the scientific progress, and some of that had happened in Oxford. So one of my mentors here in Oxford had been involved in identifying the gene for Duchenne muscular dystrophy, had been involved in identifying a very closely related gene. There was a lot of that work happening here. So there were multiple reasons to home in on that rare disease, and that was really the absolute focus. But the approach we took was an RNA-based therapeutic approach, which was oligonucleotides. And so over time, that has become the core area of expertise that I have and that my team has. And because in those days, we were immediately thinking about how do you translate this into a therapy, that took me down the routes of thinking about, de-risking early stage therapies, clinical translation, working with companies, spinning out new companies, all of the kind of things that would be absolutely necessary to take discovery from the lab and to have an impact on patients. And as a result, that led me into kind of leadership positions in Oxford, where I'm responsible or have been responsible for 10 years for building the kind of innovation ecosystem around the university here, particularly how we de-risk early stage drug discovery, how we spin out new companies, how we work with biopharma. So that's been an element of my job for over 10 years here. But based on that early experience that I've had since the 1990s of trying to do that very, very specifically around RNA therapies for muscular dystrophy. So it's been a fantastic journey. I mean, Oxford is a truly innovative translational medicine center, one of the best in the world. And a lot of that has been built up over the last 20 years, and it's been thrilling to be part of that. But, there's so much work to do in the rare disease space. I'm really trying to focus the next 10 years of my life on really making as much progress as possible in new therapy development for these rare genetic diseases, which we've learned quite a bit about over the last 25 years.
You're there at the Oxford-Harrington Rare Disease Centre. Tell me a little bit about the centre. What's the goal? How do you work on rare disease? Are they working inside labs there, or are you actually working with biotechs outside?
Yes, but all of that. I suppose the origins of this were a lot of the work that I've described to you already, but in parallel, similar kinds of work happening in the early 2000s by many people in the rare disease field, as these diseases have started to become tractable and it has become possible to think about therapy. And we discovered here in Oxford, probably just over 10, 12 years ago, that we had somewhere between 300 and 400 people in Oxford working on rare diseases. And we didn't really know that there were all these people. It's a big place, but they straddled multiple disciplines, which is essential for drug development and translation, through chemistry, through engineering, through computing, through all the obvious biomedical departments and disciplines here. But we have clinical centres, major sort of clinical centres within our UK health system exist here in Oxford. It turned out there were 300 to 400 people, and we started to bring them all together to start to work collaboratively and to build a rare disease effort within Oxford. And what we decided was that, we had this incredible research engine here, and probably one of the best in the world, and certainly probably one of the biggest in Europe. But that what we really didn't have was the ability to turn these fantastic kind of research discoveries into treatments. We didn't have that experience. That's normally in industry. That's where you find the expertise to turn a discovery into a new drug. And that's the route that ended up leading us to the Harrington Discovery Institute in Cleveland, in the US, who were doing exactly this. They had built a therapeutic development center at the Harrington Discovery Institute that was primed with ex-industry drug developers to work with academic teams to develop drugs originating from excellent academic science. And so we partnered with them. We formed our joint transatlantic research rare disease center called the Oxford-Harrington Rare Disease Center. And the mission, our goal is absolutely focused on therapy development. Our mission is to develop new treatments. We've got a very ambitious target over the next 10 years to do that. And so it's quite unusual for an academic enterprise that's sort of within an academic center to be focused on drug discovery and drug development. There are reasons for that. And, one of the biggest reasons is that industry really hasn't been involved enough in the rare disease space. And we've had to think about how we would do that with an industry mindset, but within academia, at least starting within academia. And so that's the raison d'etre of our center. It really is therapy development, but in a highly collaborative, transatlantic way to try and work with the very best people we could find, including, in industry across, multiple countries.
So are you working then with top pharma partners? Are they getting involved? Are they funding or collaborating with you?
We're working with a few. I think we are in a position where we can start to think about bigger collaborations and partnerships. The model that we have essentially is to de-risk early stage drug development in rare diseases. And I can say more about that, but it's really to do that because I think the perception in industry is that this, for many of these diseases, there's no obvious commercial model. For many of these diseases, it may be too risky. If we can really do that early stage de-risking in a really robust, scientifically rigorous way, we then have assets that we can position for partnering with pharma. And that early stage de-risking we do with philanthropy. So we are really have been generating large amounts of philanthropy that can drive the early stage drug development. And many of that is coming from foundations, from patient foundations, patient organizations, individuals. But that can underpin our early stage drug discovery development, de-risk those early stage assets and put us in a strong position to partner with industry. Now, our colleagues in Cleveland at the Harrington Discovery Institute, they had a very successful partnership with Takeda. That was before our rare disease center really got off the ground, which was about 5 years ago. But we now have a pipeline of assets, which is getting close to 50 or more assets, which are still pretty early. So most of them, not all of them, but most of them are still in sort of late preclinical. So they haven't yet got to the clinical stage. But I think we're now building up strength in a number of areas that I could talk about, but rare heart muscle diseases, but particularly rare brain and rare cancers, where we will be looking for partners to work with. And, we're in discussions at the moment with a very big pharma partner about potentially working on some of the rare brain diseases that are not obviously of interest to them, but it could be a very viable route for them to de-risk some of their internal platforms for some more common brain diseases, going down the route of a genetically well-defined rare disease as a route to de-risking a technology or a particular therapeutic platform for a common disease. They're interested in the rare diseases too, but I think the big pharma companies are obviously interested in the big markets and the big common diseases. But rare diseases, I think there's been hope and promise for some time that they could be a route to de-risk, to accelerate drug development for common diseases. And I think that's what we're certainly finding as part of this discussion we're having now with a potential big pharma partner. So I think we're positioning ourselves in a good place to do that. But I think some of that we will end up doing ourselves. We will end up raising investment. We will end up spinning out our own companies. But I think we recognize that, that working with pharma partners who know how to do this and particularly get phase 2 onwards and get drugs properly developed and accelerated through the clinical trials is what we need to do. And yeah, it's a priority for us. Absolutely.
One of the reasons we wanted to have you on is, tackling rare disease is something we've covered at Labiotech a fair bit over the years. There's obviously a need there, like you say, but commercially it's smaller than other areas which manage to win a lot more of the headlines and a lot more of the investment, at least commercially. If we start from the idea, like you do, that there is a need for these rare disease therapies, that there's a patient need there, and that the faster we get these therapies out to patients, the better it's going to be. What are the challenges that people are facing? Is it regulatory? Is it technical, financial, scientific? Or are they just challenges of getting people to imagine what's actually possible, what we can actually do now?
Yeah, I think it's all of that. It really is. I think the need, the way that I tend to describe the need now is, if you think of an individual rare disease, there are often very few patients. And these days, with our technologies for diagnostics and genomic technologies, occasionally there's only one patient. It's a very rare disease. But in aggregate, across the planet, there are 500 million patients, 500 million patients suffer from these diseases. And we're coming on to talk about platforms and how we can accelerate through platform development. That's where we could really have a very big impact on thousands, hundreds of thousands of patients, because the need is vast. And many of those patients are at the severe end, and the severe end often affects children. And so, there really is a need to be able to accelerate treatment, because, people are suffering and people are dying. And actually, there is, for many patients, a really urgent need. So acceleration has to be part of the way that we solve this problem. But I think you're right when you have asked about the challenges, because they are diverse. I mean, one of them is commercial, in the sense that maybe 10% of rare diseases are kind of commercially viable, where a biotech or a pharma company would take an interest because there really is a market and there really are enough patients. But that means 90% aren't. And we have to really think about how we make more of those 90% commercially viable. That's part of what we're doing at the Oxford-Harrington Centre through our de-risking, taking away that early stage risk. We can make some of these more commercially attractive for investment and for industry. But there needs to be new commercial thinking about this, and we could perhaps come back to that. So that certainly is a challenge. And to solve all of these diseases, we'll need to think about how we do that, because currently there are too many diseases that just are not commercially attractive. I think one of the really exciting things of being in this field now is that at least many of the scientific challenges, I wouldn't say are completely solved, but many of them are solved. The advances in genetics and genomics of the last 20 years has meant that we can identify the genes that cause most of these diseases. And most of these diseases are caused by an error in a single gene. So that's a huge advance over the last 20 years. People wondered, what's the Genome Project all about? Well, 20 years later, we've got the fruits of that. And in parallel with all of that, there have been a whole plethora of therapeutic technologies which have been developed, and to many of them quite substantially de-risked over the last 20 years. So we're sitting at this really opportune moment with a golden opportunity here, where we can identify the genes. We've got the therapeutic technologies. Of course, there's more science to be done, but for many of these diseases, the prospect of developing a therapy is actually not that difficult. It can be pretty straightforward. So I think then we come to the regulatory. I think we may talk about this further, but I think there are definitely regulatory challenges, because I think the regulatory systems, both here in the UK and Europe, and probably also in the US, were built 60 years ago for the days of small molecule drugs and large patient populations. And we're doing exactly the opposite, highly tuned genetic drugs and small patient populations. So there's real need for regulatory reform. And the regulators, and particularly here in the UK with our MHRA regulator, I think there's a real appetite to innovate and to reform. But I think I'm really pleased that you made that last comment about imagination, because I think this is really what's needed. And I think, I think we're starting to imagine what the world could look like in 10 years if we're successful. Could we develop an oligonucleotide drug or a gene editing drug? Could we do that in 100 days for a patient? Could a patient come into the hospital, get their genetic diagnosis, find out what the gene is, come back in less than 100 days, and the treatment is there? And we're starting to plan out how we could do this. Could we really have a revolution where a treatment could be developed that quickly for an individual patient? And we believe we can do it. I think most of the building blocks are there, but it requires the imagination to think about how you would do that, how you would join all the dots. It requires a lot of investment. We'd have to persuade some people to take a risk and put some money into this to prove it could work. But I think there really is a need for imagination on a large scale, because it's a vast problem. And, that's why I think it's one of the things that's really encouraged me to move away from the muscular dystrophy field, or at least to broaden out through our rare disease center, because there's a big challenge. There really is a big challenge. And we need to work with some people who really want to imagine what the new world could look like in 10 years. So I think all of those things are challenges, but I think there are solutions, and I think it's an exciting time because those solutions I believe will happen and materialize in the next 10 years.
One of the things that could make it a lot faster is just to accelerate, make things modular, make things scalable. I mean, I feel like I'm talking just in computer technology here, but it's the same terminology. We want to move faster, make it modular, scalable. What does that mean in the context of rare disease? Are we talking platforms?
Yes, absolutely. We are. So I think probably the most obvious place to think about this is with a therapeutic drug like an antisense oligonucleotide, which most people will have heard of, but this is a small piece of DNA-like chemical material that has the specific capability to home in on the genome on a single gene, on a single very specific part of a gene that might carry a mutation or an error. So it's a gene-specific or genome-specific treatment. There's a lot of chemistry in the oligonucleotide, but built into that is a sequence. And therefore, if you want to go from one gene to another gene, in theory, all you do is change the sequence. The chemistry of the drug could look identical, but it would have a slightly different sequence, and it would then be, if you like, repurposed for a different gene. Now, these types of treatments, were first described in the 1970s, so they've been around for 50 years. The first treatments or experiments for muscular dystrophy, and I've been working on oligonucleotides for muscular dystrophy. The first experiments were really reported by Japanese scientists in the mid-1990s, so we've known about these drugs for a long time. There are now probably close to 20, I would guess, perhaps more, approved by regulators in the US and in Europe. So these types of therapies really have matured to the point where there are approved drugs, where we know exactly how to develop these drugs. There are a range of different chemical variants of these drugs that have slightly different properties. And what that means is actually oligonucleotides are quite diverse in what they can do. Some of them can switch on genes. Some of them can switch off genes. Some of them can modify the way that genes are processed. And so they can be deployed in a range of different ways as platforms. So you could think about an oligonucleotide platform that would switch off a gene and develop it for one gene and repurpose it for another. You could think about an oligonucleotide platform that would switch on a gene and do the same thing, repurpose for multiple genes. So these are truly platforms. The technology is mature. There are many approved oligonucleotides, and they are a highly suitable technology for many, many rare diseases that are caused by errors and mutations in genes. Not that every rare disease would be amenable to an oligonucleotide therapy, but a very high proportion are. But do we really want to have to go through the process of developing 10 or 20,000 separate drugs when the drugs all look very, very similar? And they may even be intended for the same organ in the body. They may be developed for liver diseases or brain diseases. So you'd be taking a chemically very, very similar drug for a muscle disease and then having to go through the process, starting from scratch, to develop a very, very similar drug for a related muscle disease. It just doesn't make sense. And I think the barriers to really scaling up and really exploiting something like oligonucleotides as a platform have been, developing the technology, de-risking the technology, getting it approved by regulators, having lots of examples of approved drugs that actually work, that are safe. And I think probably one of the best examples of an oligonucleotide drug that works and is safe is the one called nusinersen, developed by Biogen and Ionis for spinal muscular atrophy. I mean, this is transformational. These kids who would die age 1 or 2 don't die anymore, and this drug keeps them alive. So we know these drugs can work, and in the rare disease context, and there's a huge and growing body of safety data. So then you have to have a conversation with a regulator to say, we want to develop these drugs as a platform. and accelerate this and massively amplify this, can we think about how you might regulate these drugs differently? That's a conversation we've been having with our regulator here in the UK. I think there's similar discussions with the FDA in the US. I think we're moving a little bit more quickly in the UK, but there's such a body of data. Can you, when you develop a new oligonucleotide, access all the safety data for the existing drugs? And if you're developing a drug that's exactly the same chemistry, do you have to go through all the safety, all the toxicology, all the animal studies that could take you millions of dollars and several years before you have a drug that you could test in patients? Or could we do things differently? And we believe we can do things differently. And so I think this could really be a highly scalable platform system where you could repurpose one oligonucleotide after another. And I think this really, really hits home when you think about some of the patients who could benefit where there is only a single patient. And you and certainly many of our hopefully our listeners in industry will know of a colleague of ours who actually, through the Oxford-Harrington Rare Disease Center, we have invested and funded and supported his work. This is Tim Yu in Boston Children's Hospital, who was the person and colleague who developed the first oligonucleotide drug for a single patient. And this was a desperately ill patient. And so there's urgency. But if we're going to have to start at square one and do all the safety, all the toxicology, go down a traditional drug development route for that single patient, the cost will be enormous, the time will be too long, the patient, will not be alive to see any benefit. And I think it was that landmark piece of work by Tim that really said, we have to think differently about this, because there are lots of patients out there where there is urgency and there is just not the commercial route to be investing money to develop drugs for individual patients. So the platform could be applicable at that very ultra rare end of the spectrum where there really isn't a commercial model, but we need to do things faster and more cost effectively. But the platform can equally be applicable at the more common end of the spectrum. And, these types of drugs are also applicable to common diseases. And the more rapidly we can build these scalable platforms, this will benefit not just rare disease patients, but patients with common indications that could also benefit from these types of therapies. So I think there is enough data, there is enough knowledge of a platform like this now, whereas something else, perhaps like gene editing, we're still learning, maybe it's still a little bit early, but it equally could be a platform. But for oligonucleotides, and I think, so I could say more about it, but certainly in the UK, there's a lot of willingness to really be innovative about how one can facilitate this from the regulatory point of view, as a true platform to benefit these patients.
Let's talk just a little bit about gene editing while we're here. Tools like CRISPR, technologies like CRISPR. What ideas do you have for scaling that when it comes to rare disease?
Yes. So I think, again, there's been a landmark development last year. Again, colleagues that we work with here in Oxford, but this was, I think, again, something the listeners will be aware of. The gene editing team in Berkeley and San Francisco, working with clinical scientists in Philadelphia, developed a gene editing therapy for a single patient. Now, I think there are more challenges for gene editing currently. We don't have the same body of data of thousands and thousands and thousands of patients who've benefited. I think there's still some safety concerns, and particularly if one has to go down the route of using a viral system for delivery of the gene editors. I think there are safety concerns in relation to viruses and viral vectors, and so there's probably more work to be done to develop gene editing as a true platform, but it absolutely could be. If you talk to colleagues of ours like Theodore Ernoff at Berkeley in California, he really believes this is a platform. Where I think there may be some difficulties is that the precision of gene editing makes it eminently suitable for the single patient, for the patient with the very specific mutation. And so I think it will be a perfect platform for these very rare cases where you really want to de-risk, where you really want to correct an error through base editing or prime editing. And that therapy may, in the end, only be applicable to a very small number of patients. It's very easy for oligonucleotides as a slightly different platform to see how these could be applied to many of the bigger indications, large numbers of patients. Whether that turns out to be the case for the gene editing platform, but it is almost the perfect platform to correct the individual genetic error that will affect different patients. I also think I've alluded to the need in many cases to use viruses to deliver gene editing. I think the gene editing is, whereas oligonucleotides, we can think about those more as a chemical type of drug. Gene editing is a complex therapeutic, and delivery is, it's a challenge for oligonucleotides, how we get those delivered to the right cells and targets in the right tissue, but it's equally a challenge for gene editing. So currently, one of the areas that we are particularly interested in is how you would develop delivery capabilities for gene editing in the brain. And that's not feasible really yet. And unfortunately for rare diseases, about 60 to 70% of all rare diseases affect the brain. So we aren't really in a position where we could deploy gene editing as a platform to tackle rare brain diseases as of today. We're very interested in the Oxford-Harrington Centre in platform development, developing technologies that could unlock that area of gene editing within the brain. That we're not there yet, but I believe that will happen within the next 5 to 10 years. But because it is a more complex therapeutic, the delivery is absolutely a critical question that needs to be solved, which is why much of the progress has been made on blood or in liver, where the threshold, the barrier for delivery of the gene editing complexes is somewhat lower. But it's a wonderfully precise genetic tool, and it's absolutely in that platform space. So we're kind of, at least in our work, we're working with ASOs and oligonucleotides, which could be a platform today, tomorrow, and certainly also with gene editing that will be a platform in the future. We hope for rare brain diseases and many other diseases, but isn't quite there yet today, I believe.
One of the other technologies that always gets brought up when we're talking about accelerating development is artificial intelligence, AI, these computational tools. Do they have a place to play there with rare disease as well? Are they something you're already applying?
Yes, absolutely. So I think alongside our Oxford-Harrington Rare Disease Centre, we have been fortunate, at least here in the UK, in Oxford, to attract a large amount of government investment into a big new research centre here in Oxford, which, if you like, sits sort of upstream of our Rare Disease Centre. It's a much more of a basic technology and research centre that can underpin new therapies, whereas the Oxford-Harrington Centre really is focused on developing those as treatments. And then this new centre is called Therapeutic Genomics. And so one of the angles in which computation and artificial intelligence, I think, is already playing a big role is just in exploiting genome data and not just identifying mutations and patients, but actually identifying new ways in which one could think about how you would correct genetic errors. And so if we really want a platform like oligonucleotides, can we use artificial intelligence and analysis of genome data to help to point us to where in the genome we can deploy those oligonucleotides? And there are lots of obvious places we've known about for a long time, but there are many newer types of targets within the genome. So that's one angle, and that work is happening. But I think what your question is kind of alluding to is, can we actually use this type of machine learning AI in therapy development itself? And, I think there's certainly been progress and, many efforts to date in terms of using AI for small molecule drugs. But could it really be used to accelerate the development of genetic drugs like gene editors or oligonucleotides? Now, I think one of the barriers for oligonucleotides, and certainly a barrier for groups like ours, groups in academia, groups like the Oxford-Harrington Rare Disease Centre, is that we just don't have access to huge amounts of data that you would need to drive an AI platform on oligonucleotides. And we were accumulating as a field huge amounts of data, but often a lot of the data that we would need on efficacy and safety of these drugs resides in pharma companies. And that data is not necessarily accessible to the clever people we have working with us here developing AI tools. But we have published something very, very recently, which is our first big effort in this space, which is really to start to extract data on oligonucleotides from the patent databases. So we don't have data in our hands on hundreds of thousands of oligonucleotides. That would be, if we could do those experiments, we'd need hundreds of millions of dollars. So we don't have it. But that data exists in the patent databases. And so we've recently, with colleagues of mine here in Oxford, have made a huge effort to extract patent data and to use that data to train AI models for oligonucleotides. That data is now in the public domain. There's an AI tool called Oligo AI that we've reported recently. And we've shown that using that tool, we can fast track the identification of a lead oligonucleotide molecule over the standard screening approach. We're now in the second stage of that work, which perhaps is the much more important stage, which is to see whether we can extract safety data. So the first step was really potency and efficacy, and we believe we've made a reasonably good start at building an AI tool that could accelerate the identification of potent, effective oligonucleotides. But I think a real game changer would be if we could start to make progress on how you would identify oligonucleotides that are also safe. And that work's in progress. We know pharma companies are doing similar sorts of things. So I think there really is a role for AI to help us. And I think that in combination with thinking about how you could really develop massive, higher throughput screens, because I don't think it'll take us away from the need for screening a drug, but I think we can probably do that in a much more rapid, more intelligent, more sophisticated way with the use of AI. So, yeah, absolutely. I think it can save money. It will speed up the process. All of that will be good for patients. All of that. So, I believe there's a real place for it. And again, I think the next 10 years, it'll be embedded as a really fundamental tool in our drug discovery work, I think.
Let me ask a few questions about this future, these 10 years in the future they're looking forward. What are the breakthroughs that you see coming in rare disease over the next 5 or 10 years? Is it going to be around ASOs? Is it going to be around gene editing, or is there something else that's just emerging that's got you a little bit excited?
Well, I think one of the really attractive things, but one of the challenging things in the rare disease field is that nothing is ever in one place. Because these diseases are rare, the patients are in one place, the therapeutic technology somewhere else, the clinical expertise, the people who can run the trials are somewhere else. There's a need to join up all of these dots. And so I think some of the breakthroughs, are going to just simply come from joining up the dots. So I'll give you an example. We know, for example, that oligonucleotides now, and this is certainly true for the one I mentioned for SMA that's approved, some oligonucleotides approved for muscular dystrophy, but we know that they don't necessarily work as well as they can or should, or as well as we would like them to. And often one of the challenges really is that we, particularly in the clinical trials, we're giving these treatments to patients who are already sick. So can we then, as we get more safety data, as we have approval from a regulatory agency for a drug, can we now start to backtrack? Can we start to identify the patients pre-symptomatically who would benefit? And one of my colleagues here has been leading the work here in the UK, my colleague Laurent Servet here, in newborn screening. So I think one of the really exciting things will simply be taking a drug that we already know works, but doesn't work particularly well, but finding the patients at birth through a newborn screen who would benefit from that drug. And so instead of waiting for these patients to get sick, we will treat them when they're well. And I think the ability to identify those patients through national genome screening programs. So we're not really doing this at the right scale in the UK, and the UK is one of the most advanced places in the world in terms of population genomes through our Genomics England effort. We've sequenced between 100,000 and a million genomes of our population, but we haven't quite got there yet with newborn screening. There is a pilot program ongoing here. There are other countries doing similar things. So I think we will get to the point relatively soon where you will be able to screen at birth for certain disorders, where there is a drug. And instead of treating a child who is sick, age 8, who will only get a marginal benefit from that drug, we would treat the child age 6 months, and that will have enormous benefit. And that will be almost a cure. We hesitate to use the word cure. We always use the word treatment. And so I think what we will see is many more patients benefiting, but many more patients benefiting significantly, where the disease is below the radar for most or all of their lives, and that will constitute a cure. So I think that is one of the big advances that we'll see. I think on the more scientific technical side, I think some of the advances will be just about how we deliver these types of drugs. We could take the current gene editing, we could take the current oligonucleotide drugs, but currently we give large doses, in many cases intravenously, to patients with safety as a major concern, because we don't and can't deliver these drugs very effectively to exactly the right cell in exactly the right organ that drug needs to get to. And there's some really exciting advances in delivery technology. A company like Avidity Biosciences, acquired recently by Novartis, that has a core oligonucleotide delivery technology which targets something called the transferrin receptor. There will be other emerging examples like that. So I think we will get within the next 5-10 years to a place where we can start to think about really precise targeted delivery of these types of drugs, which will mean lower doses, higher safety margin. Initially, it will probably mean higher cost to the drugs, but I think as this becomes embedded, there'll be ways of reducing cost of goods and overall cost. And I think the rare diseases is the place to do this, because we have a defined genetic target. We know exactly which cells to get to. We'll then do exactly that, but in the newborns. We won't do that first in the newborns. It'll be in the older patients. But that type of technology will then be applicable to patients with common diseases. So whether that will happen in the next 10 years, we'll have to wait and see. But I struggle to think that a company like Novartis has bought Avidity without thinking that technology is going to be applicable to treating certain common diseases in the future. So I think you can see this progression from treating more patients who are healthy at birth and preventing that rare disease, more targeted, highly specific delivery technologies, and then doing both of those things in the future, perhaps not within 10 years, in patients with common disease, screening, identifying who would benefit from a lipid-lowering siRNA drug, and treating them early, before they get, high levels of lipids in their blood. It makes complete sense, but it may take more than 10 years to get to that point. But I believe, early treatment and highly specific delivery of those rare disease drugs, that will definitely happen in the next 10 years. And I really am hopeful that within the next 10 years, we will be thinking about how we accelerate development of these types of drugs through scalable platforms. That will mean we can develop drugs within 100 days. Now, that's a bit more of a speculative hope, but I think there's an increasing amount of effort and discussion and planning for how we might think of doing that. So, I'm really optimistic that, we will be able to treat healthy patients with very specific drugs, and we may even be able to develop those drugs within 100 days. And I think the world would look very different in 10 years if we could do that.
Matthew, we started off our conversation. You told me a little bit how you got into neurology and neuroscience in the first place. For someone who's hearing this and is getting excited, what sort of advice do you have for them? How can they dive deeper into rare disease and maybe even come and work for you there at Oxford?
Yes, I mean, I think it's not for everyone. I think, but so many people, in science and in medicine, ultimately think about how they can make an impact. How can their discovery, how could their work, make an impact on patients, on society, on the economy, on whatever it might be. For most people, it becomes more than just about the single scientific discovery. It's, how could that really benefit patients? And I think we're at this incredible point in history where, for the first time ever, we could potentially cure these diseases, which throughout human history, 300 million years, we've never been able to cure. We've never been able to do anything about them. And I think about these patients with muscular dystrophy, which is actually at the common end of the rare disease spectrum, for whom, we're just now beginning to develop these treatments that have an impact. But for the whole course of human history, there's been nothing. And so I think this is what I would say to people. You can really now make a difference. We know how to develop these drugs. We can find the genes. There is huge opportunity to accelerate development, to get your discoveries really to benefit patients. Many of those will have an economic impact as well, because these therapies, drive job creation, new companies. But at the core is the patient. And I think somebody starting out now, in a way that I couldn't really 30 years ago, somebody starting out now could really have a realistic prospect of firsthand turning a discovery and a treatment into something that really benefits tens, if not hundreds, if not thousands of patients. And I think ultimately that is one of the most satisfying things that could come out of scientific work. It's not just about the science. It's really turning that into something which has value for society and humanity. And, this is really, really possible now over the next 20 years. And, I would encourage anybody who would like to do that to get into this field. I think you can really, really have an impact. And there are 500 million patients who need us to do this. So there's a pretty good reason to do it, but it's entirely possible. So now is a great time to be involved.
What's the best way for people to keep up with your work there at Oxford-Harrington? As it happens, we're really working to be much, much more proactive in how we communicate with everybody that we work with, through patients, foundations, academic centers, industry, government. So we're going to be really, if you like, ramping up how we communicate. But, we have websites, we have social media, we have all the obvious things that people can keep up with. But of course, Rare Disease Day is coming up. It's a special day every year on the 28th of February, where we can celebrate the advances and the progress we're making collectively as a community, as a field, in progress on therapy development. And I think that will be a particularly good time for people to hear about the work of the Oxford-Harrington Rare Disease Centre. We will make a number of special efforts, webinars, videos, etc., that will coincide with that day. But I think we are keen to keep communicating with people through our website, social media, and we will have a lot of announcements, I think, this year that hopefully patients and industry will be interested in. And I'm happy to talk to anybody anytime about our work. I think we're making a lot of progress, and I'd like people to hear about it. Matthew Wood, thanks for joining us today on Beyond Biotech.
It's a pleasure, Dylan. Great to talk to you.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.